G'day, welcome to Navadia. How are you going, everyone? I saw an interaction online last week and someone was a bit confused about mutations, so I decided to explain the mechanism behind the mutations and what they mean. This also complements my video on PCR a bit, so please check it out. Links, of course, are in the description below. Also, before I start, a quick reminder that I am selling my soul to the algorithm, and I request that you help me to do so by liking and sharing this video and subscribing to my channel. 'Twould be awesome if you did so, and of course, I thank you for it. First of all, I'm going to do a bit of a breakdown on what DNA is and its structure. If you're pretty much up to date on DNA, please skip to this timestamp here. Somewhere. Come back! What is DNA? It is a molecule that is used by the body to store information. It is found in the nucleus of the cell, hence nucleic in the word deoxyribonucleic acid. It is made up of an alternating sugar and phosphate backbone. There are 4 nucleotides: adenine, thymine, guanine, and cytosine, and they are attached to the sugar molecule. I won't talk about sugars in this video, but they are definitely more than a sweet substance you put in your morning coffee. There are 2 types of nucleotides: pyrimidines and purines. Pyrimidines look like this, while purines look like this. As you can see, the purines have an extra carbon cycle here, but of course one of the carbons has been replaced with nitrogen. The easy way to remember which ones are purines is to think pure as gold, where adenine and guanine are the purines. Fun fact for the non-chemists out there, Ag is the chemical symbol for gold. If you look at the purines, you can see that they're slightly longer than the pyrimidines. This is important to recognize because pyrimidines link to purines. This helps keep the DNA molecule a consistent width. Also, T pairs with A because there's 2 places where they can use hydrogen bonding to link, while C and G pair because there's 3 places to link. I remember the way they pair by looking at the shapes of the letters. T and A only use straight lines and C and G use curved lines. I don't know if that makes any sense to you, but that's kind of how my brain works. Also, C and G rhymes with 3, so that's how I remember the number of bronze. I hope that helps. I'm also going to do a quick rundown on how DNA is replicated because that's where the majority of mutations occur. Be warned, it is really complicated. I'm going to do the simplest version I can in the limited time I have, but be warned, this is going to be so simplified it may end up being almost incorrect. I strongly recommend you read into it to yourself because damn, it's interesting. But before I start, if you skip to this bit, I just want to point out that uracil is swapped with thymine in RNA, so the machinery in the cells know if they're looking at RNA or DNA, which I think is kind of cool. The first thing that happens is the DNA is unwound by a protein called helicase and breaks the DNA into 2 single-stranded DNA molecules. Then DNA primase creates an RNA primer which tells the DNA polymerases to start synthesizing it by taking free-floating nucleotides from the surrounding area. Topoisomerase is a protein that helps relieve tension on the DNA strand by making a cut in the DNA which allows it to unwind and then reintroduces the twist after the DNA is duplicated. There are a lot of proofreading enzymes that avoid mutations, but as we know, this isn't always perfect. I have linked an excellent video that breaks down replication with moving pictures in the description below. Remember how I said DNA is like a way to store information? That information is used to create proteins. Think of it as a set of instructions on how to build something. When the DNA is needed to be used to create proteins, it goes through a process called transcription and translation. The DNA is transcribed into mRNA or messenger RNA. It comes from reading a strand of DNA in a similar process as replication, but instead of being made into a second double-stranded DNA, it's kept as a single-stranded RNA. It is processed so it can travel to a ribosome, which is where the RNA is transcribed. A ribosome is a little bit like a machine that reads the mRNA and translates it into amino acids. I'm going to add a quick clip here from Khan Academy, which explains it a much better way than I ever will be able to.
So let's zoom in a little bit on a ribosome right over here. And there's a couple of interesting actors. One, as you can imagine, is the ribosome itself. And it is made up of proteins plus ribosomal RNA. So in the video on transcription, we're already familiar with messenger RNA, and we often view RNA, like DNA, as primarily encoding information. It's acting as a transcript for a gene, but it doesn't have to only encode information. It can also, so it's proteins plus, that's not a T there, this is a plus. it can also provide a functional structural role, which it does in ribosomal RNA. And this big, this looks like an oversized hamburger bun or something right over here. This is a super oversimplification of what a ribosome looks like. And I encourage you to do a web search for image searches for ribosomes, and then you can get a more appreciation of how beautiful these structures are and how intricate they actually are. So this is the site, and you can broadly think of the ribosome as having this, this is the top bun and the bottom bun, and it's going to travel along the mRNA from the 5' end to the 3' end, reading it and taking that information and turning it into a sequence of amino acids.
Links, of course, are in the description below. The ribosome reads the mRNA in 3 nucleotide batches called a codon. As it reads it, it recognizes each codon as a code for a particular amino acid, which are the building blocks for proteins. I'll explain this in a bit more detail later. These amino acids are then joined together to create a polypeptide chain, which then ends up folding into proteins. The ribosome looks for a particular set of nucleotides, which is AUG, or adenine, uracil, and guanine, which is what is called a start codon. It starts reading from this codon and stops reading it when it encounters one of 3 other codons, UAA, UAG, and UGA. These codons produce methionine, which is the thing that tells the ribosome to stop making the amino acid chain. I strongly recommend watching the entire Khan Academy video I've linked for more information because it is actually pretty mind-blowing. I'm not going to explain any more about DNA transcription because this is already information heavy and I don't want you to be bogged down too much. Remember, this is possibly the most simplest of simplest explanations on something that is ridiculously complicated. So I'm going to start talking about mutations. But first, what is a mutation? It is a change in the genetic material of an organism. There's 2 different types of mutation, which are chromosomal, which of course affects the entire chromosome, or point mutation, which affects one point in the DNA. For this video, I am going to concentrate on point mutations. Mutations can be found in gamete cells, such as sperm and eggs, which will end up being passed down to following generations, or in somatic cells, which aren't passed down to further generations. That is why you don't get your parents' skin cancer, but you can be prone to getting skin cancer depending on your parents. The story of the 2 girls in China where they were genetically engineered to resist HIV will have this mutation in their germ cell line, which is very concerning for scientists because they will pass their mutation on to their children. This is one of the reasons why the scientific community heavily criticized the scientists that did that experiment, among other things as well. Point mutations occur when DNA has a mistake in the sequence of nucleotides. They can occur in many different ways, but are most commonly due to environmental factors. In eukaryotic cells, such as ours, they don't occur very frequently because they have proofreading mechanisms. However, some organisms, such as viruses, don't have proofreading mechanisms, which means that they mutate at a much faster rate. For example, HIV has no proofreading mechanisms, and an HIV-positive person can have several quasi-species develop in their bodies over their lifetimes. That is one of the reasons why scientists haven't been able to produce a vaccine or a cure for HIV, and why you need to get your flu shot every year. I'm going to rewind a little bit and talk about codons. Remember how I said that the ribosome reads 3 nucleotides and goes, aha, these 3 nucleotides want me to code for this particular amino acid? An amino acid is the building blocks of protein. Protein isn't exactly just muscle, but are also used in the body for things like structure, such as your bones, certain functions, such as hemoglobin, and enzymes. Enzymes are proteins that speed up chemical reactions and lower the amount of energy required for this reaction to occur. Enzymes end in ASE or A's, and their names explain exactly what they do. For example, lactase is the enzyme that breaks down the sugar lactose. A protein structure determines how well it functions. If the wrong amino acid is put in the wrong place, the protein will either not work or have decreased function. So it's pretty important that the correct amino acids are in the right spot. If you're wondering why the weird hamburger-shaped protein ribosome reads in sets of 3 during mRNA transcription, it's because of something called codon degeneracy. This term is used not because the codons are wanting to stab you in a dark alley and steal your wallet, but because it refers to a single amino acid being encoded by more than one codon. There's 22 different standard amino acids, 20 of them being common and 2 being rare. The rare ones are selenocysteine and pyrrolysine. If the codons use one nucleotide during transcription to code for an amino acid, there would only be enough for 4 amino acids to be transcribed. If it used 2 nucleotides, there would only be enough for 16 amino acids to be transcribed because it would be 4 different possibilities in the first part of the codon and another 4 in the second. However, using 3 codons, it means that there are 64 possible permutations, meaning there's enough to make sure that all amino acids are accounted for, and there's a little bit of leeway if there is a point mutation in the DNA. For example, if you look at this chart, the amino acid leucine is coded using the codons UUA, UUG, CUU, CUC, CUA, and CUG. So if a point mutation swapped the first U in UUA with a C, the amino acid leucine will still code. This particular mutation is called a silent mutation because even though there is a change in RNA or DNA sequence, it still codes for the same amino acid. And that is why evolution is so slow. Not only does DNA have proofreading mechanisms, it also has codon degeneracy, and not all mutations are in the germline. There's several different types of mutations, and the first one I want to talk about are transitions and transversions. Transitions occur when a pyrimidine is swapped out with another pyrimidine or purine with purine. Usually this happens due to tautomeric shifts. Remember when I said G and C had 3 hydrogen bonds and that's how they selectively bond with each other? Well, a tautomeric shift happens when the hydrogen jumps to another part of the molecule. So suddenly, the guanine, for example, only has 2 hydrogens to bond with, which means it will bond with thymine, not cysteine. Transversions occur less often due to that size difference I mentioned earlier in the video. They swap a purine with a pyrimidine, or vice versa. The next type of mutation I want to talk about is frame shift mutations. They add or delete a base pair in the DNA sequence. Remember how I said codons read in 3? Well, if you add or delete a base pair in a DNA sequence, the whole thing is thrown completely out of whack. Frame shift mutations cause diseases such as cancer, Crohn's disease, and cystic fibrosis. Frame shift mutations aren't limited to one base pair being removed or added to the genetic code. Small insertions or deletions of 20 base pairs or less account for nearly 24% of known mutations. Often an insertion or deletion is caused by something called strand slippage. What happens is a strand of DNA being replicated with repeating nucleotides bubbles out and reattaches itself back in line. When the DNA is replicated again, the new DNA sequence will either lose or gain these nucleotides, depending where the template or synthesized strand had the slippage. Another type of mutation is depurination, where a purine buggers off from the DNA strand and the DNA repair mechanisms have to guesstimate whether the purine was a T or an A, and usually it inserts an A. Another type of mutation I want to talk about is a missense mutation. Missense mutations come from the swapping out of one amino acid with another due to a transition or transversion. It doesn't delete or add a nucleotide pair, it just swaps out an amino acid. Often this doesn't make too much of a problem because amino acids can be close enough chemically to not make much difference on how the protein folds, but other amino acids can cause huge differences. I'm going to link another video to Professor Dave, who explains it in much better detail than I am. Examples of diseases that are caused by missense mutations are sickle cell anemia and a rare genetic disease called progeria syndrome, where the patient ages quickly. Sadly, those with this disease often do not live beyond their first 15 years of life. A similar mutation caused by a point mutation such as a frame shift, transversion, or transition is a nonsense mutation. This happens when the stop codon occurs prematurely in the sequence. A stop codon is UAA, UAG, and UGA, and it does basically what it says on the tin. It stops the ribosome from translating any more of the protein. This means if it stops early, the rest of the protein doesn't get coded. Examples of a disease caused by a nonsense mutation is cystic fibrosis or thalassemia, which causes a type of anemia. Remember when I said there were those 2 rare amino acids? Selenocysteine is coded by the stop codon UGA, and pyrrolysine is encoded by the stop codon UAG. The reason why they are so rare is because there needs to be special mechanisms in place for the ribosome to read it as a normal codon and not a stop codon. I thought that was pretty damned interesting. I'm sure this is all fascinating for you, but what about mutagens? Can I get my spidey senses? Actually, I wouldn't suggest that because mutations change the shape of proteins, and if they aren't silenced, the chances are they're not going to be good. Yes, you can get positive mutations such as being able to produce lactase into adulthood, but as a general rule, mutations are not a good idea. Some chemicals called base analogues, such as 5-bromouracil, are mutagenic to replicating DNA. They are called base analogues because they are analogous to the molecule they are replacing. As shown by the name, 5-bromouracil replaces uracil, and it increases the frequency of tautomeric shifts where the hydrogen bonding is placed in a different part of the molecule. This causes guanine to pair with it rather than adenine. Another type of mutagen that targets replicating DNA are acridine dyes. Acridine dyes inserts itself between the stacked base pairs and increases the rigidity of the DNA molecule and changes the conformation of the double helix. That is why ethidium bromide, the chemical I talked about in my PCR video, is considered to be a mutagen. There's other chemicals that don't give a damn about whether your DNA is replicating or not, and they're called alkylating, deanimating, and hydroxylating agents. Deanimating chemicals doesn't stop your DNA from moving, but what it does do is it changes an amine group into a ketone group, and that causes effects similar to a tautomeric shift. So adenine will bind to cysteine rather than thymine, and vice versa. Guanine will still bind to cytosine, but it will only bind in 2 places rather than 3. Alkylating agents attaches an alkyl group such as methyl, ethyl, or propyl groups and changes the base pairing properties. They include transitions, transversions, frame shifts, and chromosome aberrations. They can also cause chromosome breaks. So, yeah, not the most friendly of chemicals. But how do we figure out what is a mutagen or not? Well, bacteria to the rescue, as they usually are. The Ames test is a way to figure out if there is a mutagen in the product. What happens is scientists use a mutant version of a salmonella microbe. This is auxotrophic for histidine. An auxotroph is an organism that can't produce its own essential nutrients and must get it from their environment. For example, humans are auxotrophic for 9 amino acids, and we need to include them in our diets or we will get sick. That's why you keep hearing about essential amino acids and whole proteins. Anyway, these salmonella microbes are grown on a petri dish that doesn't contain histidine. Sometimes they'll reverse back to making their own histidine, which suggests a mutation. If the chemical they're testing is a mutagen, you will see more colonies of the salmonella than you would on the control dish. Pretty simple, quick and cheap test. I could talk about other mutagens like radiation, but this video has already gone for way too long. Please check out the videos I've linked below for more information. I strongly recommend it. It is a fascinating subject. Again, if you enjoyed this video, please like and share the schmozzle out of it. I hope you have a lovely day, evening, night, whatever, and don't forget, scandium, iodine, nitrogen, and cerium. I also have to say a very special thank you to my wonderful patrons, especially my $10 Redback Spider patrons, Aided Furball, Lauren Hart, Mary Civitano, Amanda Vogue, Ross Devereux, and the Atheist Pastor. I also want to say a very special thank you to my $35 Irakanji patron, Osman Cometh. I hope you really enjoyed this. It's a really fascinating subject.